The overall goal of the following experiment is to test the metastatic abilities of human cancer cells by tail vein injections in immunodeficient mice. The cells are prepared for injection. The cells are then injected into immunodeficient mice, which will generate lung metastases. After 1 to 2 months, the mice are dissected and lung metastases are quantified and cultured. Results of this type of assay indicate the extent of metastases of a particular type of cancer cell.
Hi, I'm Sonali Mohanty from Dr. Lei Xu's lab in the Department of Biomedical Genetics at the University of Rochester. Today we will show you a procedure to perform experimental metastasis assay. We use this procedure in our lab to study cancer metastasis. So let's get started.
Begin with cells grown to about 70% confluency. Aspirate media from the plate and gently wash once with 1x PBS. Next, add 2 ml of 0.05% trypsin in Versine. Gently rock the plate to facilitate cell detachment. Observe the cells under a microscope as they detach. Add an ample volume of media containing FBS to quench the trypsin activity. Then transfer to a 50 ml tube. Count the cells using a hemocytometer. Once the concentration has been determined, centrifuge at about 200 times gravity for 3 minutes. Without disturbing the cell pellet, carefully remove the media.